So I am Mark, a PhD student in EDVAPS at Hospital Clínic of Barcelona, working in the FIBROTARGET consortium. And I'm going to talk along with Victoria about omics-based annotation, specifically about the technologies of single-cell RNA sequencing and spatial transcriptomics. So what we're going to do is explain a little bit how we use these technologies to actually decode the transcriptomic architecture of gut fibrosis. I'm going to start talking with a brief introduction of transcriptomics. So it is basically the high throughput quantification of the RNA pool in tissues and cell cultures. So by measuring RNA, we can see exactly what's going on in a tissue. So not all measurements are created equal. So depending on the technology we use, we get very different levels of resolution. And to explain this, I'm going to use an example with La Sagrada Familia, which is a really famous touristic building here in Barcelona. So imagine you want to understand this building. So first we have bulk RNA sequencing. So this basically measures the average gene expression across an entire tissue sample. So in our analogy, this is like taking the Sagrada Familia, crushing it into fine powder, and analyzing the pile. So you can tell me, okay, this contains 40% stone, 10% glass, and 50% cement. So you know the ingredients, but you have lost the structure. Next, we have single-cell RNA sequencing. So this basically measures the gene expression of thousands of individual cells. So now we are not crashing the building, we're carefully taking it apart brick by brick. So we can say, okay, here's a red stained glass window piece, here's a brick, here's a support beam. So we basically have a perfect catalog of every component, but we don't know if that stained glass belongs in the facade or in the crypt. So we have the parts, but we don't have the blueprint. And finally, we have spatial transcriptomics, which basically maps gene expression while preserving the location in the tissue. So this would be like looking at the finished building. We see the stones, we see the glass, and crucially, we see exactly where these pieces are located relatively to each other. So we are able to understand the architecture. So now let's have a look at single cell and spatial transcriptomics. So the goal here in single cell is to capture the unique profile of every single cell, right? But how do we actually do it at once? Because we need to grab each tiny cell at once and look at them, right? So we generally use, well, there are generally two main strategies. So the first one is droplet-based technology. So this basically involves encapsulating a single cell and a barcoded bean within an individual oil droplet. So effectively, its droplet functions as an isolated reaction vessel, and this allows for single cell processing. This is a technology that has been developed by Tinex Genomics. And then we have sequential barcoding, which is another type of technology for single cell. So here, instead of trapping the cell, we use the cell itself as the reaction vessel. So if we pass a series of chemical cocktails over the cells, building up a unique molecular ID tag or barcode inside each cell. And this has been developed by PARS. So now that we have a little bit knowledge of the different technologies of single cell, let's talk about spatial transcriptomics, which is currently the cutting edge technology. So as I said before, this allows us to measure gene activity without losing the architecture of the tissue. And we usually classify the spatial transcriptomics into two methodologies. So the first one is the sequence-based method, which functions by partitioning the tissue into a grid of microscopic squares. So we sequence the genetic material from each individual square, where the resolution of our data is directly determined by the size of the squares themselves. So this approach enables us to capture the whole genome across the entire tissue slide. However, because we are working at the square level, many RNA molecules are basically ligated into a single point, providing a high density snapshot of that specific spatial location. And in contrast to this technology, we have the image-based methods, which we basically keep the RNA exactly where it lives. So how does this work? So we basically apply fluorescent probes that find and bind to specific genes, which we then visualize using high-power microscopy. And a major advantage here is that the molecules are not ligated to a single point or pixel, and they remain free in the coordinates, right? So this means we are not looking at an average of a square, but rather the precise location of every single molecule. So as you can imagine, this generates incredible amount of data, and it is very multi-layered and very complex. So red data comes into many strange formats in single cell and spatial, depending on the technology that you are using. But ultimately, our goal is to actually convert all this data into gene expression count matrix. So this table that we see here is basically a count matrix, and it is crucial to store gene expression data. And we obtain it from the raw gene expression data that this technology gives us. As you can see, this spreadsheet is basically a table where rows are the genes and the columns are the cells. And the numbers inside tell us how many times a specific gene was active in a specific cell. But that's not all, because I said it's multi-layer. We also have different data. So we have the metadata information about the sample, in which it stores the patient age, the conditions, etc., depending on your study and your work. And also in spatial, we have even more layers, which can be high-resolution images of the tissue, coordinates of molecules, polygons of the cell boundaries, etc. So how do we handle all these complexities? So to handle that, we rely on specified coding containers. That's how we call them. And there are many packages available that you can work with to actually put all this data together. If you work with Python, one of the most common ones is the undata object. And if you work with R, also one of the most common ones is the Seurat object. These basically act like digital suitcases, keeping the count matrix, the images, and the metadata all together and organized. And finally, once we have processed our raw data, how do we actually make sense of all of that, and how do we analyze it? So the analysis pipeline usually follows a standard logic, but it always changes depending on every data set and on the technology that you're working on. So the first thing that you need to do once you have processed the raw data is perform quality control. Here, we basically filter out the damaged cells or empty droplets, cleaning up all the noise. And we have different ways to actually perform the quality control. We can look at the cells themselves, see how many counts they have, how many genes, if they have high percentage of mitochondrial genes, if those cells are behaving as tablets. We can also look at the features, seeing how they are expressed across the entire data set. And also in spatial, we can look at all the spatial layout, where we can look at the coordinates and see different spots and if the data is working properly. So after doing that, we have to perform normalization. So technically, we need to know that different cells might have more or less RNA just by chance. So we use math to level the playing field so we can compare them fairly. Then we have dimensionality reduction, because analyzing, for example, 20,000 genes at once would be very challenging and also very computational demanding if you are working with a lot of cells. So we use dimensionality reduction, and this helps compress the dimensionality of the data for further analysis. And once we have simplified our data, we can perform clustering. So this can be unsupervised, where we ask the computer group the cells that look similar, or supervised, where we tell the computer what to look for. And yeah, this basically helps us identify cell types that we are going to talk about in the following slides. And I think it's also important to mention that each workflow is basically aligned to your data set and your technology. And what we've seen with our experience is that every analysis is very different and very complex depending on what you're looking for, the data that you have, and the tissue that you have. Now, Victoria is going to talk about the clustering.
Okay, so I'm Victoria Budiño, a postdoc at Azucena Salas lab in Barcelona, and I'll continue on explaining about the omics in IBD. So once we have seen how the analysis work, we are now going to discuss how we can annotate the cell types and the applications of these technologies to study fibrosis. So as Mark has just mentioned, data can be visualized in these UMAP plots, where each dot is a single cell, and cells are grouped in clusters based on their transcriptomic similarity. So an important step before annotating the cell cluster is to decide which is the resolution that we are going to use. And as you can see here in these plots, the more you increase the resolution, the more clusters we get. And this decision is up to the researcher to make. So ideally, we need to be careful not to overcluster the object, and we should stop increasing the resolution once the clusters that we get don't have biological meaning. So to help us understand the ontology of the clusters, we can make use of this plot, which is a clustering tree. And this will allow us to see where the new clusters in the highest resolution come from, and it may be quite useful when we are trying to choose the right resolution to work with. So clusters are annotated based on differentially expressed genes from one cluster to the other. So there is a, sorry, in the screen, do you see this?
We can only see like the title of the slide, and then the slide is blank.
Okay, no, sorry, because I thought that you were seeing the other camera. Okay,
No.
sorry.
No,
okay, I will start again in this
Perfect.
in this slide. Okay, so clusters are annotated based on differentially expressed genes from one cluster to the rest of the data. And the first characterization of the intestinal stroma compartment by single-cell RNA-seq in inflammatory bowel disease was published in 2018 by James Kitchen from Alison Simon's lab. So what they did was to sequence colonic biopsies from healthy patients and patients with IBD, conducting a tissue digestion to enrich for the mesenchymal compartment. So in this analysis, they identified 4 types of fibroblasts and characterized their gene markers and location in the tissue. So as you see here, stroma 1, 2, and 3 were homostatic fibroblasts found in the healthy mucosa, whereas in inflammation, it emerged a specific inflammatory fibroblast that they annotated as stroma 4. In addition, they also identified the myofibroblasts, 2 types of endothelial cells, glia, and pericytes. So since the study from Kitchen, many other publications have sequenced the stromal compartment of the intestine by single-cell RNA-seq. And while some have kept the nomenclature of S1, S2, and S3, others have preferred to annotate clusters based on their top marker genes. So as you can see, we currently don't have a standardized nomenclature for the intestinal fibroblast, but still, we see quite a consistency of the subtypes that we normally identify. No, for instance, here in the study from Kitchen, the fibroblasts S2 were annotated as Win5b in the study from Ms. Bailey, and as NPY positive cells in the study from Kong. And if you see the transcriptome of these cells, they are all the same cell type. So potentially, the more samples you sequence, the more cell types or states that you can identify in your single cell object. But there are a number of clusters of fibroblasts that you can almost always identify, regardless of the number of patients that you include in your dataset. So now we will have a quick overview of these cell types and which are the main markers that of genes that you can use for the annotation of these cells. So as mentioned earlier, and as identified in the study from Kitchen, there are mainly 3 types of fibroblasts in the healthy mucosa. So we have first the fibroblasts S1 or APCA-A and ADAM-DEC positive cells that are those in the lamina propria that we have in here in blue in the drawing. In contrast, we also have the fibroblasts S2, that are those that underlie the epithelial crypt, and they are subdivided based on their location. So there are the S2A, which are at the bottom of the crypt and overexpress BSTM2A or SOC6, and there are also the S2B, which are the ones at the top, and they overexpress NPY and neuregulin. And finally, there are the fibroblasts S3 that underlie the crypt bottom and express stem niche factors such as R-responding or GREN1. So on top of this, in the healthy mucosa, we can also identify fibroblast reticular cells or FRCs, which are within the lymphoid aggregates, expressing the lymphocyte recruiting cytokine CCL19 or CCL21. There are also the smooth muscle cells that separate the mucosa to the submucosal layer and express myosin 11 or ACTA2. Regarding endothelial cells, blood vessels are identified by the von Willebrand factor and PKAN1, while lymphatic endothelium specifically expresses LIFE1 or MRC1. We then also have the pericytes that are the cells surrounding the endothelial cells, and they are easily identified by RGS5 or Notch3. And finally, we can also commonly see glial cells by single cell studies that they express neurexin-1 or S100P. So altogether, this is the cellular architecture of the healthy mucosa, and inflammation will happen. So in contrast, in inflammation, what we see is a reduction of the number of these homeostatic fibroblasts and then the emergence of new inflammatory cells. So there are mainly two types of cells that they emerge in inflammation. There are the inflammatory fibroblasts that express a battery of inflammatory genes such as the MMPs 1 and 3, interleukin 24 or 11, T3L1 or tenascin. And then there also appears a new type of endothelium, which normally is annotated as inflammatory endothelium, that mainly express ACKR1. So this is regarding the annotation of single-cell data. But what happens with the spatial transcriptomics? So we can apply essentially the same type of markers, but the annotation works is slightly different due to an intrinsic limitation of the technique. So as Mark has explained already, in sequencing-based technologies, that is where the RNA is captured by especially barcoded probes that are bound to the slide, more than one cell type may be captured with the same spatial barcode, and therefore what we will get is an average expression of different cell types. So even though now the resolution of these techniques is increasing, it is still challenging to obtain a single-cell resolution with sequence-based technologies. And in contrast, in image-based, this relies on cell segmentation. And we know that cell segmentation, of course, is not perfect. So RNA from different cells may be analyzed as the same cell. So because of this challenge, when we are annotating spatial transcriptomics data, it is recommended to choose low resolutions and also to be aware that a mixture of genes from different cell types may be observed when within the same cell cluster. Okay, so once we have seen an overview of how to work with this data, we will now have a quick look at some examples of how can they help us to investigate intestinal fibrosis. So an example of single-cell RNA-seq are these studies from members of our consortium and Thang and colleagues. So these are two independent studies, and they both conducted single-cell RNA sequencing in intestinal resections from patients with Crohn's disease, covering non-inflamed, inflamed, and also stenotic regions. So when they conducted single-cell analysis of the stromal compartment identified a subset of fibroblasts that were specific to inflammation and fibrosis that they both annotated as Fab fibroblasts. So pathway analysis indicated that these cells were enriched in extracellular matrix and collagen processes, and they validated their increased abundance in fibrosis by flow cytometry. So moreover, staining of Fab positive cells indicated that these fibroblasts were mainly localized at the submucosal layer, that is where extracellular matrix normally accumulates in fibrostenosis. And mechanistically, they observed that the transcription factor TWISC1 is induced in Fab fibroblasts, and they functionally test that this induction can be promoted by macrophage-derived cytokines, so altogether forming a profibrotic signaling hub of fibroblasts and macrophages. In contrast, now regarding spatial transcriptomics, fewer studies have yet applied spatial to study fibrosis, but an example of such is this study from Kong and colleagues, where they generated a single-cell and spatial transcriptomics atlas from paired stenotic and non-stenotic regions. So they conducted differential abundance analysis, and it reveals an increase of myeloid, plasma cells, inflammatory, and also cells from the follicle. So this observation, together with the results from the single-cell analysis, highlights that in fibrosis we can have an expansion of immune-rich regions and also an expansion of tertiary lymphoid structures. So apart from that, they also identified two types of fibroblasts that interestingly seem to have antagonistic locations in the tissue. So on one side, the so-called inflammatory fibroblasts, which are located closer to the epithelium, and they seem to associate to ulcerated areas. And then the other type of fibroblasts are the ones that they annotated as collagen-high tissue fibroblasts, which seems to be located at the deeper layers of the mucosa. So interestingly, these later, the collagen high, they express FAB, which altogether is in line with the findings that we just saw from single-cell studies. So even though the inflammatory fibroblasts are far from the stenotic foci, they are far from the submucous and deeper layers, a recent study from the same group highly suggests that they could also be relevant in fibrosis. And what they did in this study was to integrate single cell and also spatial transcriptomics data. And by doing so, they characterize the transcriptomic signature of these inflammatory fibroblasts, which highlights that they have an important inflammatory, but also a tissue remodeling role because of the expression of all these collagen genes. So given the spatial colocalization of the inflammatory fibroblasts and also together with activated macrophages, they functionally validated the cellular signaling circuit involving the activation of the transcription factor GLIS3, and they identified that this factor is necessary to promote a pro-fibrotic and also pro-inflammatory gene program in these fibroblasts. So altogether, we can state that single-cell and spatial transcriptomics are powerful technologies that can help us to create an intestinal cellular map in health and disease. They can also help us to identify pro-fibrotic mediators that could potentially be targetable in the clinic, and also they help us to understand the intrinsic heterogeneity of this disease. Thank you so much.